Yeah, thanks. So if you don't know, my name is Brian Callam. I have taken over directing the Red Dial, or the Louisiana Sea Grant Oyster Research Laboratory from John Supan, my predecessor, which is on Grand Isle, Louisiana, and is located at the Michael Wazan Oyster Hatchery. Our hatchery, we run it through a cooperative partnership between Sea Grant through LSU and the Louisiana Department of Wildlife and Fisheries. So if you're ever down in South Louisiana, stop on by. This presentation today, I'm going to do a little bit of results of some stuff we've done in our hatchery, which follows really well with what we just saw from Kelsey. And then talk a little bit about some of the activities that we're doing and that we're going to be doing moving forward. So to jump into it, in the hatchery, we're really interested in seeing how the things that go on in the hatchery line up with things that have to happen on the farm. We're also interested in, as a hatchery, trying to do whatever we can to make improvements in production efficiency inside the hatchery. Small gains in the hatchery are going to result in more seed coming out, better seeds coming out, and better products at the end when you harvest them off the farm. And because in our region, triploids are important, we're looking at mostly traits in triploid study. So we wanted to see how some of these traits are affected by their genetic background, who their parents are, does that matter? And we have two different groups of tetraploids at our hatchery. They both share common ancestry, so we weren't sure going in how different they are, whether or not we can actually consider them different populations or not. So we wanted to look, if we make triploids using these two different tetraploids, do we see anything different, or are they all the same? So we took our two different tetraploid groups, and we made those with two of our most used diploid breeding groups to create 4 different types of triploids. And we looked at some of what we consider relevant, important hatchery traits. And these are things like hatching rate. So how many of the fertilized eggs survive to the first tank drop on day 2 at the D stage? Once they get to that D stage, how many of those actually survive to setting? It doesn't matter if you have 100% hatching rate, if they all die before you set them on anything. And then, of course, just the size of the larvae and harvest proportion. So this is essentially what Kelsey just presented on. How many early setters, how many peak setters, how many of those late setters, and of course the duration of that harvesting. We think this could be important for improving the production efficiency in a hatchery. If you can harvest an entire brood over 1 or 2 days rather than stretching it out over a week or a couple weeks or any given time, you're going to improve your production efficiency. So we looked at all these traits and saw how their parents affected those traits. And what we found was the hatching rate and this harvest proportion, how many come off on a given day, both of those are affected by which of these tetraploids we use and which of the diploids, and to some extent the combination of those. So looking at hatching rate, so where the cursor is, the top two, 4NA by 2NA, and 4NA by 2NB. So those are the triploids we made with one of our tetraploid groups. The hatching success for that was around 50%, which was higher than what we saw from the triploids from a different tetraploid group, which had lower and more variable hatching success rates. So for the hatching rate, we can see that if your dad is from one of these tetraploid groups, you're going to have a better hatching performance. And then for the second group down at the bottom, if you're from this second tetraploid group, it depends. It's important which diploid we're mating with those tetraploids to get your triploids. So we're seeing that the selection, triploid larvae aren't all the same. It matters who the parents are, both the mother and the father. So that's for the hatching rate. How many of the eggs hatched out into the swimming larvae? But what's interesting, so there's differences here, but then the ultimate survival to the eyed larvae, to the settling part, was the same for all the triploids. So the same amount of larvae coming through at the end to set. So maybe this, in practice, how well these hatch out doesn't really matter if you have enough eggs moving forward. So looking at the harvesting of the petty villagers, we saw a difference again. They separated out mostly by the tetraploid that we used, so who your dad was. The closest ones on the left, the yellow and that green line, are from that second tetraploid group. This is the group that had the lowest hatching rate. And you can see down at the bottom where it's days post-fertilization, that's when we started harvesting these eyed larvae out. So the groups from the tetraploids that had the lowest hatching rate, we also started harvesting eyed larvae out of those groups early, around 10 days, which was pretty great for us. When you can see the second group, the purple and blue lines, we didn't start harvesting those until about 2 weeks into the culture period. So you can get a little bit of difference in production efficiency if you harvest out most of them a few days early, right? But those ones that harvested out early, you can also see that those peaks aren't real narrow, tight peaks. So we're harvesting early, but then the harvest continues on for a long time. We're getting a lot of those late setters coming out. But in practice for a commercial hatchery, most of those late setters probably would have terminated the group. You know, it might be 50,000 larvae that we're harvesting off of a day. Probably more to keep going, and rather you're just doing a respond and put things back up. But the groups, the blue and the purple lines, those are the ones that had the greatest hatching rate, again, so the same parents. And we see a short harvest duration, so we're able to get the development to the eyed larvae seems pretty synchronous. They're all happening at the same time. And we can get those larvae out of the tanks over a short period and then get new larvae into them. Which is nice if you're receiving eyed larvae from somebody. You'd rather have the whole order all at once rather than having it split out and trying to harvest over a week. So the next steps for this part is going to be doing these process again to make sure it's repeatable and it wasn't just something the conditions we hatch over time. And then also looking at how they grow out in the nursery and how that we grow out in the field and see if any of those things match up. So other activities that we're doing and that we're going to be doing in our hatchery and on our research farms. In the hatchery, we're going to be looking at water quality. It's really important for any hatchery, but especially in our location, in the part of the Gulf we are, we have extreme variation in conditions. So salinity, temperature, DO, all these types of water quality things that are important swing very widely over very short periods of time. So we're tracking these closely. We're going to be working with some of the other commercial hatcheries in the area and some of the research hatcheries and see if we can line up when conditions change at our site. Is somebody else seeing that, and then are they seeing the same dips or peaks in production in their hatchery that go along with that? We're also looking at doing some probiotic treatments in our hatchery. So we have a couple of studies we're going to be doing this year. Hopefully at the end of the year we'll have some information to share with everybody on this. Looking at probiotic use, we've seen a lot of bacteria fluctuations in our hatchery. So we're looking at things like just general bacteria cocktails that you might dump into your hatchery water. Or what I think is really cool are these things called balos, which are predatory bacteria that eat Vibrio bacteria up that you can possibly put in there. So we'll have more information on that towards the end of the year. We're also really trying to nail down conditioning of broodstock in our portion of the Gulf Coast, especially tetraploid. We don't have a really good handle on exactly what it takes to get those animals in spawning condition. We have a good handle on, once they're there, how to hold them at spawning condition for a pretty good chunk of time. But getting them to that point so far has just been a bit of black box. We looked at some and they're in the spawning condition, great, and hopefully we can get more into it. So we're really going to try to nail down a protocol and a formula on how to exactly do that. And with that comes some work we're doing with a cryopreservation group to scale up cryopreservation to a commercially viable scale. And we can have, importantly, tetraploid sperm, but really any sperm available at any time. There's some hatcheries in Florida that come online extremely early for our broodstock already. So if we can't get this conditioning part down, that may be a way of getting around that issue. In the nursery setting, any setting is really part of the hatchery too, but small gains in production efficiency are really going to magnify out, especially when we talk about our need for seed in the region. So if we can bump up 10% setting efficiency with just simple manipulations of the setting procedures, that would be great. So we're looking at things like that. And then also improving seed through the rearing process, not just developing and improving the nursery technologies, but also how we're operating those. We've also heard some concerns from some of the growers in Louisiana that we have a lot of freshwater input at the beginning of the year. And that's also the beginning time where we're putting out a lot of seed from hatchery, but they can't take it because it's 2 parts per 1,000 on their farm, and all that seed can die. So we're going to look at ways maybe we can harden the seed for some of these stressful conditions, or hold the seed back in a practical way until they can actually get it. And then on the farm, we're always looking at improving our triploids and our diploids. So extending that larval comparison out into the farm and seeing how the tetraploid's appearance are going to affect the triploids. And as always, we're constantly looking for new wild populations in our area to see if we can have any of those characteristics come into our breeding groups that make an oyster more suitable for a farm at a given site. And this wild population testing has really taken off, especially last year after all the freshwater. We went out and collected a lot of animals from areas that went on to experience significant mortality, 90 to 100% in some of these wild populations. To try to save and maintain that genetic structure, and then we can incorporate that into some of our breeding things. I also have clams up here. It's something that we're looking at on the farm. There's been on and off efforts of mostly one individual at LSU to get to do work with rangium, freshwater clams, and they're just extremely abundant in Louisiana. They don't taste very good. And it's geosmin is what it is, it tastes muddy. So we're looking at things, diversifying your oyster farm with maybe these freshwater clams, if you want to bring them in and decorate them in more salient conditions, and see if you can actually get them to taste decent. So we've been doing this. We've done a little bit of pilot work on transplanting and decoration, and it looks promising. We're able to get the geosmin levels extremely low. And the survival from transporting them from Lake Pontchartrain, which was 0 parts per 1,000, to our Grand Isle site, 20 parts per 1,000, the mortality was lower than I expected to observe. So that would be interesting to see how that goes. And then, of course, the economics behind that, once we decide if they can actually handle this type of thing. So those are all of our activities. We're also going to be opening up our order forms as one of the providers for a seed larvae in our state. So if you're interested, that's going